In this episode, we'll explore inherited disorders. We'll start by outlining what inherited disorders are, then discuss examples such as cystic fibrosis and polydactyly, and finally look at embryo screening and the ethical questions it raises. Some disorders can be inherited from your parents through the genes they pass on to you. These genetic conditions are caused by mutations in specific genes that lead to health problems. Let's start by looking at cystic fibrosis, which is a genetic disorder affecting cell membranes. It results in the body producing a lot of thick, sticky mucus in the air passages and in the pancreas. Cystic fibrosis is caused by a recessive allele. This means that for a person to have the condition, they must inherit two copies of the recessive allele, one from each parent. Because it's recessive, people with only one copy of the allele won't have the disorder. They're known as carriers. For a child to have cystic fibrosis, both parents must be either carriers or have the disorder themselves. As genetic diagrams show, if both parents are carriers, there's a 1 in 4 chance that their child will have the condition. Now let's look at polydactyly, which is quite different in terms of how it's inherited. Polydactyly is a genetic disorder where a baby is born with extra fingers or toes. It doesn't usually cause any other problems, so it's not life-threatening. Unlike cystic fibrosis, polydactyly is caused by a dominant allele. This means the disorder can be inherited if just one parent carries the defective allele. As the genetic diagram shows, there's a 50% chance of a child having the disorder if one parent has it. Modern medicine now allows for screening of embryos for genetic disorders. This process is done during in vitro fertilization. In IVF, embryos are fertilized in a laboratory and then implanted into the mother's womb. Before being implanted, it's possible to remove a cell from each embryo and analyze its genes. Many genetic disorders can be detected in this way, such as cystic fibrosis. It's also possible to get DNA from an embryo in the womb and test it for disorders. There are lots of ethical, social, and economic concerns surrounding embryo screening. Embryonic screening is quite controversial because of the decisions it can lead to. For embryos produced by IVF after screening, embryos with bad alleles would be destroyed. For embryos in the womb, screening could lead to the decision to terminate the pregnancy. There are strong arguments both for and against embryonic screening. Those against embryonic screening worry that it implies people with genetic problems are undesirable. This could increase prejudice. They also express concerns about where to draw the line. Could parents eventually select for traits like eye color or intelligence? Others note that screening is expensive and question whether it's a good use of resources. On the other hand, supporters of embryonic screening argue that it will help to stop people suffering. They point out that treating genetic disorders costs the government and taxpayers a lot of money. Some also believe there are limits to how far screening should go, but at the moment, parents cannot even select the sex of their baby unless it relates to a health reason. Understanding inherited disorders and the technologies available to detect them raises important questions about the intersection of science, medicine, and ethics. While advances in genetic screening offer new possibilities for preventing suffering, they also challenge us to consider difficult questions about what kinds of interventions are appropriate and where the boundaries should be. In our next episode, we'll explore sex determination and how the combination of chromosomes determines whether a baby will be male or female.